Okay, let me share the screen. We'll hop into it. It should just be one slide deck for this week. Boom, there we go. Okay, so there are a number of characteristics of cells in life. What makes cells cells, right? Why viruses aren't cellular, why viruses aren't alive. There are certain characteristics, right? They can be one cell organism, most like bacteria or single celled, or they can be multicellular organisms like us. But regardless of the number of cells, there are common denominator characteristics that are shared among these cells, a shape of some sort, whether it's circular or cylindrical or like a cube. It's going to have a, a defined shape that maybe changes, but comes back to its own self. It's going to have what's called cytoplasm, the stuff, the liquid sloshing around on the inside. And then around that cytoplasm is going to be the membrane. It's going to have nucleic acids, whether it's DNA, and then that DNA making RNA, or it's going to have ribosomes. Remember that viruses aren't alive because they don't have a cell, they don't have ribosomes, they can't make their DNA into RNA by themselves, or they can't replicate the RNA if they're an RNA virus by themselves. But cells, on the other hand, are different. They do have their own ribosomes. They do have an ability to undergo metabolism, to break down things into smaller things, right? To break down molecules into things that they can then use. And we said at the beginning of this class that there are two major types of cells, eukaryotic and prokaryotic. And what is bacteria? Which one of these? Anyone?
A eukaryotic?
We are eukaryotic because
Prokaryotic.
yes, so humans have anything that has a nucleus and a membrane-bound organelle is going to be eukaryotic. So think you do have nucleus. Pro, no nucleus. So bacteria doesn't have a nucleus. It just has its DNA arranged in chromosomes or in the circular plasmids. And so they are going to be prokaryotic or prokaryotes. When we have eukaryotes, we think of animals, plants, fungi, protists. So yes, there are some microbes that are eukaryotic. The prokaryotic microbes are the bacteria and the archaea. And again, the difference is that eukaryotes do have a nucleus and membrane-bound organelles. Prokaryotes do not have those things. Cells are going to have genetic material, right? And that genetic material is what they pass down to their progeny, as well as that material helps them program for the different functions of life that are needed to keep that cell in existence, whether it's to grow and divide and to differentiate into different things, or if it's just merely to produce enzymes and undergo metabolic processes in order to gain nutrition and subsist. Okay. Cellular organization, if it's not a bacterial cell, right? If it's a multicellular organism, cellular organization is going to give rise to the tissues, to the organs, right? You take the organs, you break it down, you get cells. And so all of these things come together to make life what it is. And so if we look at these things, there are about, what is that, 6 major characteristics that define life. Okay. One, of course, is reproduction and heredity. So in order to be alive, you have to be able to reproduce you have to have that ability to reproduce, whether it's sexually or asexually, budding off like many yeasts produce asexually. You have to grow and develop, right? That is a characteristic of life. And when you stop growing and stop developing, that usually means you're in the waning years of life. You have to undergo metabolism, right? Your body, your cells have to undergo chemical and physical processes. You have to move and or have an irritability, if you will. Okay, so that means you have to be able to respond to the environment, whether it's responding in a very mobile way, whether it's you moving because you have muscles and bones and connective tissue, or you're moving across a chemical gradient of some sort, you're being attracted via chemotaxis. There has to be some kind of recognition of what's going on externally and internally and a reaction to that, right? You have to have a cellular support, if you will. So some kind of protection, some type of membrane or coating or storage mechanism. So when we start talking about the bacterial structures, one thing we'll talk about is granules and how they're used to store things. These are all support mechanisms for the cell to keep it alive, whether it's the membrane to prevent bad things from coming in, or it's ion channels to pump bad things out, or it's storage granules to sequester certain things that in high concentrations can be toxic to the cell, like zinc. All of these things have to be present in that cell to maintain homeostasis. And then transport of nutrients and waste. It's got to be able to take in nutrients. It's got to be able to export wastes that are no longer beneficial. Okay. These are the characteristics of life. Okay. So let's spend a little bit of time looking at the anatomy of a bacterial cell. And I recommend that you become familiar with all of these components. Okay, that will be beneficial to you. Let's start with, let me get to the annotation feature here. Okay, so if we look at the top half here, what we have is the cell wall, right? The cell wall is going to be this membrane on the outside of the cell that protects it, that lets things in and lets things out, but does it selectively. And then outside of that cell wall, you have what's called a capsule. Now, not all bacterial cells have capsules. And if you remember, when we were talking about staining procedures, there's a capsule stain, right? Where if the bacteria has a capsule and you put India ink on it, it'll repel that India ink and you'll see this kind of halo around the bacterial cell. So in the very far exterior of the cell wall, you have a capsule for protection. You have the cell membrane, which is kind of hard to see. So right here, this yellow part is the cell membrane. And that's the sandwich, right? We have the cell membrane. On top of that, we have a cell wall. And then in some bacteria on top of that, we have a capsule right here. All right. There's also going to be a glycocalyx or slime layer right here. Okay, that slimy stuff. And that functions for a number of things. One of the things is to make a biofilm, which we'll talk about later. You may have heard of biofilms. It's just kind of like a network of bacterial cells that are all kind of coordinating together and have created this film based on their cells, and then this secreted glycocalyx slime layer stuff. Okay. Biofilms can form in catheters in patients and cause UTIs and things like that. What else do we have? We have these structures here called fimbriae. And you might remember fimbriae because I really glossed over it, but we talked about when we were talking about the steps for a microbe to invade. The first one is portal of entry. And the second one, what's the second step after portal of entry?
Is
Anyone?
it adhesion?
Adhesion, yeah. Adhesion or attachment. So the fimbriae are used to attach to a host cell to invade it, or just to attach to a surface of some sort. Okay, so, and then the last kind of outer structure is a flagellum. Not all bacterial cells have flagella. Some do, some don't, and some of them have different arrangements, and we'll talk about that in a little bit. But those are going to be on the outside for motility. And you can discern flagella from fimbriae because the flagella are bigger and longer structures. The fimbriae are like little like Velcro-y attachments. They're shorter. And then I guess I forgot to mention one more external structure is this pilus or pili if there's multiple ones. And those are used for conjugation, which we'll talk about later too. So those are the outside components of a bacterial cell. Not all bacterial cells will have all of these components, but these are this is the variety that is potentially there. On the inside, what we have are chromosomes, right? All this stuff, that's DNA. And note, it's not packaged into a nucleus. It's just free in the cytoplasm. What is the cytoplasm? The cytoplasmic matrix, that's this stuff, the fluid, the media that all of these things are within. The goo. There will also be actin filaments. So the actin filaments are these hair-like structures. And we have actin filaments in our cells. Bacteria has actin. And those polymerize and they allow things to be transported along them. It's kind of like a railroad track in terms of shuttling different molecules from one end of the cell to another. It's also in our cells used for motility to kind of stretch the cell out and then contract and then stretch out and contract. So actin is very important. And then finally, we have these inclusion bodies here and ribosomes here. Okay, these dots. Remember, the ribosomes are what are used for translation into protein. Okay, so be familiar with these different components of the cell, be able to label that cell if need be. Okay, two major groups of appendages. One for motility, that's the flagella, and the other one being the fimbriae that we mentioned for attachment. So movement and attachment are the appendages. The glycocalyx is the surface coating, the slime layer that's used for instance, to make biofilms, and also for movement as well. Flagella is composed or made up of three major parts. The filament at the end, it's the long, thin structure that waves and wiggles around. The hook, which is where that kind of attaches to the bacterial cell, And then the basal body, which are these rings that are within the membrane of the bacterial cell as the anchoring point. Okay. And this is what it looks like. The flagellar structure on the left is from a gram-negative bacterium. The one on the right is from a gram-positive bacterium. And those two we'll get into more detail later. But to just, I think, suffice to say, the major difference is that the gram-negative has a significantly more prevalent periplasmic space right here. It's thicker, whereas the periplasmic space of gram-positive is thinner. And as a result, it has a gram-positive has a much thicker cell wall, whereas the cell wall for the gram-negative is thinner. It also has an outer membrane, which is absent in gram-positive. But we'll get into that more in detail later. What is different between the flagella and these different types of bacterial cells is just simply the rings. Okay, so because you have this greater periplasmic space in the gram-negative, there will be a couple rings here that are embedded in the cell wall and also in the cell membrane. Whereas in gram-positive, you just have the rings embedded in the cell membrane, not in that outer membrane and cell wall. Okay? Pretty straightforward. Now, flagellar arrangements can be 4 different types. If you have just one flagellum, then you are going to be monotrichous. So here's the little bacterial cell, and you just have a single flagellum like that. You are monotrichous. If you are lophotrichous, then you are going to have a cluster or a bunch of them, but they all come from the same site. So like this. That is lophotrichous. If you are amphitrichous, well, then that means you have one on each end of your cell. So one there and one there. And then peri, that's going to be all over. So all over. All right, so these are the 4 different varieties, and you'll need to know what these 4 different varieties are. They look like that. So which one of these, okay, this one up here in the top left corner, is that going to be mono? This one here, is that mono, lopho, amphi, or peritrichous?
Monotrichous.
Bingo, because there's just one here at the end. How about this guy here? Is it going to be amphi, lopho, or peri?
Lophotrichous.
Lopho, right? Because there's a bunch of them coming out just one end. And this one?
Amphitrichous.
Amphi, yes, two ends. It can be one on each end. It could be multiple on each end. In this case, it looks like there are multiple, so it's kind of a lopho amphi, if you will. And then so that means this one is peri because they're all over the place. Good, thank you. The flagella are, again, used for movement. They rotate 360 degrees, and the direction that they move, or the coordination in which they utilize, allows them to move in certain patterns, okay? So if the rotation of the flagellum is counterclockwise here, that's going to cause it to what's called run, which means kind of just dart forward. And then how does it change direction? It rotates in the opposite way. So if it's going counterclockwise, it's gonna run, and then if it needs to turn for whatever reason, it's gonna switch the direction to clockwise, and that's gonna cause it to angle downward and move in a different trajectory. If it has multiple flagella, so in this case, this looks like it's a peritrichus, then they will all coordinate together and have this. Where's my drawing thing? What happened? Sorry. They will all coordinate together for a run, like a shoot forward. And then they will disperse and reorganize themselves and then independently on their own move in a clockwise pattern to then do a tumble. And that's how these critters get around. So they are a little bit more sophisticated than maybe some of you would have suspected. They are capable of sensing their external environment based on chemical cues and either going away from it if it's a bad cue or going toward it if it's a favorable one. What guides these responses? Well, chemotaxis, different chemicals in the milieu. Phototaxis, light stimuli, and if that's a good thing, it will go toward the light, especially if it's things like cyanobacteria that can photosynthesize. Maybe it doesn't like light because it's a sterilite, the UV is not good. It will move away from it. And again, it does this using a series of runs and tumbles. A run is going to be that linear trajectory, and then a tumble when it goes clockwise, when it rotates its flagella clockwise, is going to cause it to tumble. Okay. And so this is kind of the pattern that we'll see. If there's no attractant or repellent to guide the movement of that cell, you'll see it basically kind of just wander around aimlessly in a series of runs and tumbles, right? So here's a run, and then it does these tumbles, and that allows it to change its direction, but it's not really going anywhere. It's just going in a circular pattern. But if we have something like a chemo gradient or concentration gradient, so it's white here, low concentration, and then it gets progressively more pinkish to the right. So the chemical gradient is concentrating, and it's causing this bacterium to move. Yes, not just a full-on run straight toward it, but you can see it's making its way by doing these series of runs and tumbles to get there. The run is going to be longer than the tumble, so typically. Make sense? Okay, so it's not the most efficient way to get from point A to point B, but for a bacterial cell to be able to do that, I think is quite profound. There are some flagella that aren't on the outside of the cell. There are some that are periplasmic, meaning they're in the periplasmic space, which, if you remember, we talked about this here. Gram positive, gram negative. There's a space in between the cell membrane and the cell wall. That's the periplasmic space. Some flagella reside in there, in particular types of bacteria. And what that does is it's not, it's not waving around, flapping around, rotating around. It's twisting. And that torsion is causing the bacterial cell to ring, ring like a sponge or a towel. And as a result, the ringing and then the expansion after that contraction causes it to move in that way, kind of like a worm in a way, kind of inches its way along rather than swimming. Okay. Any questions on flagella? All right. Fimbriae, the hair-like projections. Proteinaceous, that just means they're made of proteins and they're on the cell surface here. And what they do is they allow the adhesion of that bacterial cell to its host or surface. So these are the Fimbriae right here. And sorry guys, someone's calling me. Don't know why. What happened to my screen? Sorry. There we go. And so here are some E. coli cells. This is a particular type, serotype E-Tech, which we're going to talk about the different serotypes later on in the class, in the course. But these Fimbriae are right here. And that's what allows them to attach. In this case, it's E. coli attaching to the microvilli in the intestine, those finger-like projections on the intestinal epithelial cells. Okay. And that's how they attach. And then from there, they invade. Clear the drawings. Pili. That was the last outer cellular structure. And I said it was used for conjugation, right? These are only in gram-negative cells. And what they do is these pili will attach to a neighboring bacterial cell and transfer its DNA into that bacterial cell. Okay. And this is a way to get variety. This is a way for bacterial cells to evolve and expand their genome. It's how things like antibiotic resistance is passed on from one type of bacteria to another. Okay. If a bacterial cell evolves a mutation that allows it to be resistant to a particular antibiotic, then it can pass on that DNA to another cell that doesn't have that mutation by using this external straw that pokes into that bacterial cell and injects the DNA into it. Okay. Glycocalyx, that's the slime layer, right? The glycocalyx. What is glycocalyx? Glycocalyx, glyco, sugar, and some degree proteins. And they're secreted on the outside. They have a loose organization. They don't really attach firmly to the surface. That's what the Fimbriae are for. They're just there as kind of an ooze, or, as it says, a slime that helps it move around. It also kind of protects it from things. Now, some bacterial cells also have a capsule. The capsule is, in contrast, very organized, almost impenetrable. Okay. And so the components that make up that capsule are very tightly attached, and it kind of closes off things from the outside. It also prevents its own hydration from leaking out. So a capsule benefits a bacterial cell when it's in a very desiccated environment, because bacteria is a living thing. It likes water just like we do. And if the cell dries out, that could be the end of it. So the capsule is there to protect it from harsh conditions, whether it's dry or even warmer temperatures that it doesn't like. It can be protected by the capsule.
Is the glycocalyx the same as in human cells? Or is
No.
it different?
Yeah,
very different.
it's different. It's a different kind of, I mean, it's made out of the same things, sugars and proteins, but it's not to be confused with eukaryotic glycocalyx.
Okay.
Okay, so again, it prevents these capsules prevent dehydration, prevent the loss of things that the cell wants to keep in. It can also protect it from our immune system. Okay, so remember I mentioned that some bacterial cells can be phagocytosed, but they can evade the process of, once they're engulfed by that macrophage, they can evade the processes that break it down, those lysozymes and stuff. And that is going to, one of those ways is by having this capsule. Okay, so the enzymes in that macrophage cannot break down the bacterial cell because it can't get through that thick glycocalyx capsule. Okay, also these help form biofilms. Okay, these colonies of multiple different cells bacterial cells that can communicate to each other and are almost protected by this thick glycocalyx secretion. Okay, it allows them to set up, an established colony in a host or on a surface of something that protects it. Here's a biofilm on a catheter. Okay, so here's the matrix. It's kind of hard to really see the difference between the matrix and the cells, but this right here is the catheter itself, and you can see all of these things just kind of forming a multi-aggregated assembly of these bacterial cells that then make it hard to wash off, make it hard to sterilize and treat. And so they're just hanging out in this catheter waiting to cause UTI after UTI. What time is it? Okay, doing well. The cell envelope. So this is going to be on the outside of the cytoplasm, and it's going to be composed of two different layers, the cell wall and the cell membrane, which I showed in that picture of the gram-positive and gram-negative with the flagella. And the cell envelope serves the same role as any real membrane. It's for protection, right? To give the shape of the cell and also protect it, and only allow certain things in and certain things out as necessary. And I also mentioned that we have two different types, the gram-positive and the gram-negative. And these two different classifications are by far the predominant way in which we put different bacterial varieties into their bins, right? We say it's either gram-positive or gram-negative because that tells us a lot about the membrane, okay? Gram-positive has a very thick wall, primarily made up of peptidoglycan and cell membrane. Peptidoglycan, remember, peptides, and glycan, sugar, so sugary peptides. Gram-negative is going to have an outer cell membrane and just a thin peptidoglycan layer and its cell membrane, okay? And we can differentiate the two by the gram stain, which I think is maybe next week, maybe this week or next week. One of the labsters, you'll be doing a gram stain. Unfortunately, you won't be doing the real gram stain, but that's something that is done quite frequently. And it's a differential stain, right? Because you can differentiate between two different types. And it has two colors. So there's a counter stain to it. And based on the color that's more predominant, that will tell you if it's gram positive or gram negative. Again, peptidoglycans are the primary component of a cell wall. And it's a peptidoglycan is this repeating mesh of cross-linked peptides that come together. And what that ends up doing is forming a nice barrier on the outside. Things can't get through that mesh very easily. It looks like this. Okay, so you have, and I'm not going to make you guys be responsible for knowing how these things are laid out. This is not a biochemistry class, but you have these alternating NAG and NAM cyclo rings. Okay, so NAG is N-acetylglucosamine, NAM is N-acetylmuramic acid, and so these two different rings come together in a staggering formation, and as a result form this capsule on the outside. This is what it looks like on a little bit higher resolution. We have a NAM here, we have a NAG here, and then they are linked by this bridge of different amino acids, glycines and alanines and glutamates and all these things that you don't need to know for a quiz or an exam in microbiology. But maybe you'll need to know this if you're taking organic chemistry or something, I don't know. But for now, this is just for your own information. Now, it might be helpful to know that peptidoglycans consists of NAGs and NAMs, but I'm not going to hold you to what NAG and NAM stands for. Okay. Okay, let's get into a little bit of gram positive and gram negative cell wall, and then we'll take a break, and then we'll get into our brainstorming session for our proposal. Okay. So the gram positive is going to have a peptidoglycan that is pretty thick, right? And so it's going to have a thick peptidoglycan in a very thin periplasmic space. And there are going to be different components that make up this cell wall, okay? It's going to have phospholipids. It's going to have porins. It's going to have lipoproteins. It's going to have different membrane proteins and lipopolysaccharides. The lipopolysaccharide is the LPS for short that triggers the immune response in hosts. And teichoic acid or teichoic acid. Okay, those are the predominant components of a gram-positive cell wall. And they do different functions, right? Porins are going to be the pores that allow certain things to get out, certain things to get in. Gram-negative are going to have an inner and outer membrane and much more periplasmic space, okay? Because its peptidoglycan layer is thin. And the outside membrane is going to, again, contain the same structures that this gram-positive cell wall has. It's going to have the LPS. It's going to have porins in there that allow things to enter and leave, and so forth. So this is looking at the gram-negative cell wall. And again, it has peptidoglycan, only it's much thicker. It has membrane proteins. It has porins. But what doesn't it have?
The teichoic acid?
Yeah, it doesn't have the teichoic acid. So it is different to some extent than gram positive. So there are ways that you can differentiate the two cell walls between these different varieties. Okay. So if we look here, this is just a comparison of the two. And I'm sorry, I misspoke when I was talking about the gram-positive. It looks like the gram-positive doesn't have LPS, right? I guess that key is just showing all the different components. The gram-positive doesn't have LPS. It just has the teichoic acid, which is absent in gram-negative. And instead, it has the LPS. Okay, so my bad. Sorry about that, if that was confusing. Any questions on this?
Does the gram negative not have?
Oh, sorry, I missed. I heard you up until gram negative. Does the gram negative, and then you
Sorry, does the gram positive not have porin proteins?
Oh, yeah, it looks like the gram positive does not have these porins right here. Yeah.
Is that due to the thickness of the wall or what?
Yeah, well, I think it's more due to the lack of this outer membrane. Okay, so if you look, that's a great question. If you look at the gram negative, you'll see that the peptidoglycan is right here. It's the meat of the sandwich, right? It's in between. And then on top of that, on the outside, is this outer membrane that's made up of the lipopolysaccharides and the phospholipids and things like that, and porins, of course. Okay, so here with the gram positive, you don't have that outer membrane at all, so you don't have those components to make up that outer membrane, which are, in this case, the LPS and the porins. It does have phospholipids down here, but it's below that peptidoglycan. Okay. So the main differences that we can say is that a gram positive has a thick peptidoglycan, gram negative has a thin one, and gram negative also has an outer membrane, which is lacking in the gram positive. Okay.
Makes sense.
All right, it's 10:50, so let's stop there. Any other questions, though, before we take a break? Okay, let's take a 10-minute break. And what I want to do once we return is we'll just go one by one and talk about our topics and see if we can help each other come up with some ideas for how to move the topics forward, okay? And then when it's your turn to discuss your topic, I want you to take some notes and then just turn that in. That'll be the lab assignment turn in to submit for today, okay? So let's take a 10-minute break now, and I'll see you guys at 11.